In one 125 milliliter Erlenmeyer flask, we added 5 milliliters of yeast, 2 packets of sugar, and about 50 milliliters of warm water. After we top the flask with a blue balloon, this flask will be our control flask. In our other 125 milliliter Erlenmeyer flask, we added 5 milliliters of yeast, 4 packets of sugar, and about 50 milliliters of warm water. After adding all this, we top the flask with a red balloon. This flask will be our experimental flask. Well, we let these two flasks sit for about 25 minutes, checking and comparing the circumference of the balloons with the string about every 5 minutes. Our controlled experiment was a lighter cream color, while the experimental test was more of a dark brown. However, this could have been because some of the sugar used was brown sugar. Both experiments bubbled about the same amount, but the buildup that formed on top of the controlled test was more of a foam, and the buildup on the experimental test was more chunky or curdle. The amount of buildup was also at a higher rate for the controlled test. The amount of buildup on the controlled test increased faster than the experimental test, and the balloon grew faster.
We believed that the experimental solution would create more colder because we added more food sugar for the yeast.
We believed that the experimental flask would produce more CO2 because we gave the yeast more food, so it should continue to produce CO2 longer than the control flask could.
Our experiment did not support our hypothesis. The control flask produced more CO2 than our experimental flask. In the end, our hypothesis was incorrect.